Hi, my name is Aishi, and today we'll be covering the fluid mosaic model of the cell membrane. So what is this model telling us? Well, the fluid mosaic model describes how phospholipids and proteins are essentially arranged in the cell membrane. So here we have a cell, and if we zoom in to the cell membrane, we can see that we have a bilayer of phospholipids, as shown here, and we have proteins that are either embedded within the bilayer or proteins that will attach onto the side. Now, in addition to the phospholipids and integral and peripheral proteins, we also have carbohydrates that can attach to the membrane, as well as other lipids that are not necessarily phospholipids. So we have lipids, carbohydrates, and proteins that make up the main components of the cell membrane. So why is this specific model of the cell membrane called the fluid mosaic model? Well, it's saying that the cell membrane is a fluid structure, and it's made up of a mosaic of various proteins that are embedded within the membrane. Okay, so now let's talk about phospholipids. So here we have a diagram of a phospholipid, and as you can see, it has a head component and two tails. And phospholipids are amphipathic, which means that they have both a hydrophobic component and a hydrophilic component. Now, in the phospholipid, the head is hydrophilic and the tail is hydrophobic. So the tail is made up of lipids, and the head is made up of phosphate group and another group, which is also hydrophilic. As you can see in the diagram, within the bilayer, we have the phospholipid heads pointing outwards and the tails pointing inwards towards each other. And this makes sense because this allows the heads to interact with the aqueous environment. And they're perfectly happy being close to water as they're hydrophilic, and it protects the hydrophobic tails from the aqueous environment. So this bilayer is stable. Other components of the cell membrane will also be amphipathic. For example, look at this integral protein right here. Well, the ends of the protein have to be hydrophilic, and the insides have to be hydrophobic, as you know, the inside has to be comfortable being next to the hydrophobic tails of the phospholipid bilayer, and the two ends have to be comfortable being close to the heads and being close to the aqueous environment. And if this were a channel protein, we would expect the inside of the protein to be hydrophilic as well. So what is the evidence behind the fluid mosaic model? So how do we know that it's actually a bilayer, and how do you know that they're actually proteins? Well, there's a technique you can use. It's called freeze fracture. What you do is you freeze the cell and you fracture it with a knife. And what you see is that the fracture plane will often follow the hydrophobic interior of the cell membrane. And so you get this, the membrane will split along the middle of this bilayer here. And what you end up with are two separate membranes. So you have the extracellular component here and the cytoplasmic portion here. And you can see that some of the integral proteins will go with the extracellular layer, and some of the integral proteins will go with the cytoplasmic layer. And where the proteins used to be, you'll get this little gap in the membrane. And you can see all this using electron microscopy.